Mollusks provide a very useful and easily studied means of examining how the nervous system works. The size and robust nature of the squid's giant axons made them particularly suited to the techniques available to early researchers. But today, techniques are available to study small neurons and even minute areas of their membranes. Individual neurons can vary in shape enormously, but they all have similar characteristics. A receptive zone, formed of finely branched dendrites, a processing center, the cell body, and a conductive zone, which conducts nerve impulses along the axon to its terminals. In the squid, the dendrites and cell body are located in the ganglion, and the giant axons transmit the nerve impulses which stimulate the mantle muscles. Nerve impulses can be mimicked by artificially stimulating the axon with an electrode. Experiments on a wide range of animals have shown that there's a high concentration of potassium inside the nerve cell and a high concentration of sodium outside, and that there's a potential difference across the membrane, a resting potential, with the inside surface negatively charged and the outside positive. So what are the mechanisms responsible for these findings? The large concentration gradients of sodium and potassium across the membrane of the resting nerve cell are produced by sodium-potassium pumps within the membrane, which transport potassium ions into the cell and sodium ions out. Despite the concentration gradients of these cations, the presence of equal numbers of negatively charged anions would make both the inside and outside of the membrane electrically neutral. But ions are also able to leak through the membrane, and the resting membrane is more permeable to potassium ions than sodium ions or the negative anions. So potassium starts to diffuse outwards down its concentration gradient. But as it does so, it creates an increasing charge separation at the membrane. So the layer on the outside of the membrane becomes positive and that on the inside negative. This creates a voltage across the membrane. As it increases, the resulting membrane potential retards the outward flow of potassium. Eventually, the tendency of potassium ions to move out of the cell due to its concentration gradient is exactly balanced by the tendency of potassium to enter the cell because of the electrical gradient of the membrane potential. With a negative potential on the inside relative to the outside, the membrane is said to be polarized. Although the exact methods used in these experiments need not concern us, it's clear that when the axon is stimulated, a pulse of electrical activity is generated. This is called an action potential. How is it formed? The effect of the stimulus is to introduce positive charge inside the membrane and negative charge outside. This local potential withdraws charge from the membrane, depolarizing it. If the depolarizing effect of the stimulus is large enough to exceed a threshold value, the depolarization triggers voltage-activated sodium channels within the membrane. These open, allowing sodium ions to flow down their concentration gradient into the cell. These positive ions further depolarize the membrane, which in turn triggers the opening of more sodium channels. The net effect is an increasing rate of depolarization as more voltage-activated sodium channels open. This is an all-or-nothing change and is independent of the strength of the stimulus. An action potential of fixed magnitude is always produced for any depolarization above the threshold value. But the change in the potential across the membrane slows and eventually reverses. There are two reasons for this. Firstly, all voltage-activated channels pass through a cycle. On activation, they open, allowing ions to pass. But after less than a millisecond, they slam shut again. This inactivated state persists for a few milliseconds. The sodium gates cannot be activated during this so-called refractory period, which lasts until the membrane voltage has been brought back to its resting state. The second reason is the presence of voltage-activated potassium channels. These open much more slowly than the sodium channels and are open to their full extent only when the sodium channels are in their inactivated state. The outward flow of potassium ions through these channels repolarizes the membrane. Once the membrane is repolarized, the sodium channels pass from the inactivated state to their original closed state, ready to respond to a further stimulus. The passage of an action potential causes only minute changes in the sodium and potassium concentrations, which are readily restored by the sodium-potassium pumps. Let's review this process. The response of the voltage-activated sodium channels to an above-threshold depolarization is an example of positive feedback. Progressively, more channels open as the depolarization increases, so the change promotes an increase in the rate of change. The voltage-activated sodium channels cycle to the inactivated state, stopping the flow of sodium ions, and the depolarization is reversed as voltage-activated potassium channels open, allowing potassium ions to flow outward, repolarizing the membrane. This is a relatively slow process, and the refractory period, in which the voltage-activated sodium channels are inactive, lasts for several milliseconds. This has important consequences for the propagation of the action potential down the axon. Depolarization of part of the axon causes local potentials which exceed the threshold value. So the surrounding membrane is depolarized and an action potential is formed. This in turn tends to depolarize the surrounding membrane. But the sodium channels in the area where the action potential originated are inactivated, so the action potential can only move down away from the site of the original stimulus. Since the stimulus originates at the point where the axon leaves the neuron cell body, the axon hillock, the action potential is forced to travel down the axon to its terminals. Because the action potential is an all-or-nothing response, nerve impulses are able to travel long distances unattenuated. But the on-off effect of the action potential is a rather coarse instrument and is insufficient to explain the complex behavior that's seen even in simple animals. Here, Tritonia responds with a programmed escape response to the touch of a predator starfish. Clearly, it would be an enormous waste of energy, not to say disruptive, if it responded in this way to the touch of anything. So, how can the nervous system discriminate between types of input signal? Experiments on the nervous system of the sea slug, Aplysia, show that its nerves don't all behave in the same way. In their resting state, some nerves produce no signal, while others produce regular signals without any stimulation. In response to stimulation, some neurons increase their rate of firing or fire in bursts. So, although each action potential is all or nothing, the nature of the pulsed signal with time can be very sophisticated. The change in the frequency and sequencing of the pulses enables the transmission of complex messages and provides the potential to grade the response. But to make use of such sophisticated input signals, the animal needs some sort of brain. Clearly, the connections between nerves are vital to the processing which takes place in such networks. Axons end in a number of terminals where nerve messages carried by action potentials travelling down the axon are passed on to other neurons, muscle fibres or other target tissues. These junctions are called synapses. The presynaptic ending of the axon contains many thousands of synaptic vesicles. Each vesicle encapsulates many molecules of a neurotransmitter, such as acetylcholine or dopamine. When an action potential arrives at the presynaptic terminal, it causes voltage-activated calcium channels to open, allowing calcium ions to flow down a concentration gradient into the terminal. The rise in the calcium ion concentration triggers the release of the neurotransmitter into the synaptic cleft. The postsynaptic membrane frequently contains receptors which are linked to selective ion channels. When the neurotransmitter locks into its receptor, the ligand-gated channel opens. If the ion channel allowed sodium ions to pass, the postsynaptic membrane would be depolarized and the target tissue stimulated. But some ligand-gated channels allow other ions, such as chloride, to cross the postsynaptic membrane, hyperpolarizing it. So the stimulation of the receptors linked to such ion channels has an inhibitory effect in the target tissue. Neurotransmitters simply act as messengers, so-called first messengers. The effect depends on the receptors and the ion channels to which they're linked. Indeed, in different parts of the body, the same neurotransmitter may activate either excitatory or inhibitory postsynaptic receptors. The healthy nervous system also has mechanisms for terminating the action of neurotransmitters. For transmitters like acetylcholine, there are enzymes on the postsynaptic membrane which hydrolyze the neurotransmitter, reducing its concentration and stopping its action in no more than 2 milliseconds. But not all postsynaptic receptors are linked directly to ion channels. Many receptors activate enzymes which generate second messenger molecules within the target tissue. One common second messenger system responds to the arrival of the first messenger at the receptor by stimulating the enzyme adenylate cyclase. This synthesizes the second messenger, cyclic AMP. The role of second messengers is to convert and amplify the information carried by the first messengers into an intelligible intracellular signal. Such signals can open or close ion channels or indeed trigger changes at almost any level in the cell's machinery, even to the extent of altering the pattern of gene expression. But postsynaptic receptors can be influenced by substances other than the natural first messenger. Drugs and toxins may be able to mimic the action of neurotransmitters, binding into the postsynaptic receptor and stimulating it. Substances which mimic the action of neurotransmitters are called agonists. There is another class of substances that bind into the receptors, but these don't stimulate the receptors, they simply block the normal action of the neurotransmitter on its receptors. Such substances are known as antagonists. So, in medicine, agonists can be administered to make up for a depleted supply of naturally produced neurotransmitter. Antagonists, on the other hand, can be administered to reduce the effect of an oversupply of natural neurotransmitter. In the past, most drugs have been found by screening large numbers of naturally occurring compounds and their derivatives. But today, the search for new drugs is guided by a much fuller understanding of the body's detailed structure and intricate chemistry. We've come to the pharmaceutical company SmithKline Beecham to learn about the strategies and techniques involved in such fundamental research.
Why are drug companies interested in the central nervous system?
Well, developments in basic research in all areas have preceded developments in drug development in the pharmaceutical industry, and the central nervous system is no exception to that. The exciting and rapid growth in basic research in the central nervous system has now made available the possibilities for us in the industry to develop tools to tackle diseases of the central nervous system. And these include diseases such as Alzheimer's disease, Parkinson's disease, stroke prevention, and depression and anxiety.
So what's your particular interest?
I'm interested in the function of the dopamine system in the brain. It's been known for a long time that the dopamine system is intimately involved in the control of voluntary movement. And one of the main reasons why we know this is because people who have Parkinson's disease, which is an inability to control movements, show a very marked degree of loss of dopamine-containing neurons in the brain, particularly those that project from cell bodies in the substantia nigra, in the midbrain, up to the corpus striatum in the forebrain, which in the rat is a distance of about 4 millimeters.
What's your starting point?
Well, it's been known for many years now that the dopamine-containing neurons in the substantia nigra can be inhibited by dopamine. And this is a result of in vivo electrophysiological experiments recording extracellularly from these very cells. Furthermore, dopamine will inhibit the release of dopamine from the terminals of these very cells at the point of termination of the pathway between the substantia nigra and the striatum. So what I set out to do was to try and understand the mechanism whereby dopamine caused this inhibition of firing of substantia nigra dopamine-containing neurons. And we set about doing this using an in vitro brain slice technique as opposed to the more traditional in vivo methods. I've been recording intracellularly from dopamine-containing neurons in the substantia nigra of rats in a brain slice.
The 300 micron thick slice from the substantia nigra area of the rat's brain is placed in a tissue chamber and bathed in a solution of similar composition to cerebrospinal fluid. The slice is weighted down with pieces of platinum so that it doesn't move during the experiment. The electrode can be targeted very precisely to record intracellularly from the area of the chosen nucleus. These in vitro conditions offer considerable stability and accuracy. Furthermore, the ability to control and manipulate the extracellular medium enables much more sophisticated biophysical and pharmacological data to be gathered than is possible in vivo.
What I've found is that dopamine will inhibit the firing of these cells by hyperpolarizing the membrane. And this hyperpolarization is brought about by an efflux of potassium ions from the inside of the cell to the outside. And the channels that carry this potassium efflux are activated by dopamine as a consequence of it acting on a particular receptor on the membrane. And this receptor is classified by different agonists and antagonist drugs as a D2 subtype. D2 receptors have been shown by other workers to be responsible for the inhibition of dopamine release in the striatum from the terminals of these same neurons. And it's quite possible then that the mechanism that I've just described also pertains to the way in which dopamine inhibits its own release within the striatum.
But aren't there other types of dopamine receptors?
Yes, there are. There's another one called the D1 receptor. Both receptors are found in striatum, but only the D2 receptors are on the dopamine-containing neurons. So it seems likely then that in order to understand what the consequences of dopamine release within the striatum are, we've got to look at both the consequences of activation of D2 and D1 receptors on striatal neurons. And the next series of experiments, the next program of work that I want to undertake involves recording from neurons in the striatum in a brain slice to try and answer these questions.
So how does your research help in the discovery of new drugs for diseases such as Parkinson's disease?
Well, brain slice electrophysiology essentially allows us to focus on the mechanism of drug action at the level of single neurons in the mammalian brain. As it happens at the moment, we've got a drug that's in clinical trials for treating Parkinson's disease. And these techniques will allow us to compare and contrast the action of this drug on single neurons with that of dopamine, which is the endogenous agent whose depletion causes Parkinson's disease and whose action we want to mimic with our drug. So essentially understanding the function of the dopamine system will allow us to direct and to focus the development of new drugs that are targeted at that system.
Chris, Mike works with brain slices, but you use neurons in culture. Do they have any advantages?
Well, using cultures in a way is a natural progression from the slice work in that it provides us with an even more closely defined system to work with, so that we can precisely control the environment around the single cell that we're working on, so that we can apply drugs and know that drug concentration is getting to the cell that we're recording from. And we know that we're recording from a cell that's not in contact with any other cells, so the responses we see are from that cell alone.
You use a technique called patch clamping. How does that work?
Well, in contrast to the slice work that Mike's been describing, using a conventional microelectrode where you impale a cell with a very sharp glass electrode, patch pipettes are much blunter with a polished glass tip. And the idea is to attach the tip of the glass electrode to the surface of a cell, and there it forms a very high resistance seal with the cell membrane. Now the next trick is to just disrupt the area of membrane right underneath the pipette tip, and then you have contact between the inside of the pipette and the inside of the cell. And this allows you to record membrane currents flowing through the whole cell membrane, allowing you to both voltage clamp the cell membrane and record currents activated by drugs or by changes in the membrane potential.
So how do you go about studying single ion channels?
Well, this really involves a very similar technique. We use the same glass patch pipettes, but instead of disrupting the piece of membrane under the pipette tip to go whole cell, we pull that patch of membrane away with the glass pipette, and the seal is so tight that we can do this. And then in this simpler system we can study the movement of current through these single ion channels in that patch of membrane, and this allows us to get a good idea of how a single drug molecule is interacting with a single ion channel.
We've seen some of your work on brain slices and neurons in culture. What does the future hold?
Well, this sort of work over the last 10 years has given us a much clearer idea of how neurons in the central nervous system actually communicate with each other. And knowing this gives us obvious targets for attacking disease states that up to now have been untreatable. One good example of this is therapies for stroke, which is a major killer in the Western world. And we now know that most of the damage that occurs following stroke seems to be primarily due to calcium entry causing neuronal cell death. We know a lot now about the mechanisms by which calcium gets into cells, and so there are now obvious targets for pharmaceutical companies to develop drugs to prevent calcium entering through the ion channels that we've been studying with these electrophysiological techniques. Hopefully, if we can prevent calcium getting into the cells, then that will prevent them from dying and hopefully alleviate some of the disabling symptoms that are seen following stroke.